Today is the last lecture for this course. This is a wrap-up lecture where we're going to be revisiting some of the concepts that we've learned throughout this class, touching on a few things and systems that we maybe you'd forgotten about a little bit, hopefully not. But it's all within the context of a subfield of neuroscience that I have great appreciation for, and I hope that you will too. I kind of think of this lecture as almost like a Nova special for neuroscience students. So we're going to be looking at some really kind of interesting animals and seeing how their brains do these interesting things. And really what I'm talking about is the subfield of neuroethology. So we learned about ethograms in the last lecture, right? The drawing of behavior across a day to see circadian patterns. It's because etho refers to behavior, and neuroethology is the study of the brain circuits that regulate behavior in animals. But it's a little bit more complex than that, because that's just neuroscience, right? Brain circuits drive behavior. It's with the specific context that it's an analysis of the evolutionary and comparative approach. So it's using that approach to study animal behavior and its underlying mechanistic control by the nervous system. And it's used to better understand neural control and behavior in natural contexts. So it's thinking about, how do animals use complex neural circuits to do interesting things that allow them to survive in the wild. And then, that teaches us an awful lot about how nervous systems work. It can give us additional insight just in general by focusing on the extreme examples of interesting behaviors in particular species. So we're going to look at 3 examples of this today. But first, we have already done a little bit of neuroethology in this class. So the song control system is considered to be one of the premier examples of neuroethology. And it's studying this specific brain circuit that's used for producing learned vocalizations in these animals. It's learned. We can look at it in a natural context, and we learned a little bit about that using simulated territorial intrusions. And then we also learned about the basic circuits that underlie this, the descending motor pathway in green, and the anterior forebrain pathway, which is homologous to the cortical basal ganglia thalamocortical loop in mammals. Dopaminergic input, we learned about that. So you can learn about the kind of basic reward pathways that tie into this. So it teaches us an awful lot about how these circuits work. And but this is a neuroethology system because it's used for attracting mates and establishing territories, which allows these animals to improve their ability to reproduce. We briefly talked about this a little bit. So barn owls that hunt with a combination of auditory and visual systems, and that this is a complex map of where these owls have to have a tight integrated system, which is going to regulate how they navigate space, and using 2 different sensory inputs. How do they go about doing this? How does it develop? There's been a lot of work on this in barn owls, and so we're taking advantage of the fact that barn owls have to do this really complex task, and so therefore they're going to have some interesting neural circuit that's going to reveal how that circuit works. And in addition, we can learn a little bit of how it develops as well. We also learned about the cichlid fish, the African cichlid fish, ASTATOTILAPIA burtoni, and that they have this really weird 2-part male system where most of the males look sort of female-ish and they're subordinate. And but then the dominant males have this bright coloration that they can turn on within minutes, and they show complex dominance behaviors, and that they are the ones who have all the mates. But this teaches an awful lot about how the hypothalamic pituitary gonadal system works and its basic role in sexual behavior. And we can learn how the idea of turning the furnace up and down in a very simple system where we can turn the furnace up, causing the fish to be dominant. And then, we can see how the HPG system reacts to all of that. So it teaches us an awful lot about that as well. So there are other examples, and we're going to talk about a few of these. So like I said, we're going to talk about 3 of them. You probably have heard of this system, of course, that bats echolocate. That is that they emit sounds, they are vocalizing, sound waves project away from the bat, even as they're flying. And then the waves will hit objects in space, and then they'll bounce back and be detected by the bat's ears. So there's a delay in which, from the bat's perspective, that the call will be emitted. The bat will know, of course, when it emitted the call. The wave has to travel to an object and then bounce back and then be detected. So this time difference between the call and the echo will tell the bat precisely how far away the object is. So they have brain circuits that calculate this difference and this time difference. So they can do an interoral time difference, and this is how they can then create basically a map of 3D space using sound waves by measuring this difference in detection of the call, when the call was made and when the echo is returned. So they're listening for the echo, and this has been the inspiration for inventing sonar. So a ship passing along can map the seabed by emitting sound waves. It bounces off the seabed and then is received by a transducer on the ship. There's a transmitter, which would be like the voice box in the bat. It bounces back. This is the echo, and then the transducer hears it. This is essentially the ship's ears, and the time difference detected between when it was sent out and when it was detected is going to tell the ship precisely what the seabed, how far the seabed is below, and you can map this, the depth of the ocean using this technique. The other thing that they can do with the bats, because they have 2 ears, and we learned about interaural time differences as telling you where objects are in space, they can do the same thing by listening to when an echo hits the left ear versus the right ear. So they can calculate this interaural time difference in order to determine that this object is, say, on the left-hand side versus the right-hand side, because the echoes returning to the left ear precede the right ear. And so then they can get a precise 3D space of how far it is and where it is on the left and right axis. The mustached bat, PTERONOTUS parnelli is the species name here. This is the mustache bat. Pretty crazy looking bat. It uses echolocation to hunt for insects, what's called on the wing. So as they're flying, they will be sending out signals. It'll obviously map 3D space, but it'll also be looking for prey items. And one thing that's pretty cool about these bats is that you can see is this bat has echolocated. It's seen this moth in space using its ears, and it swoops in, and it has a little pouch between its legs, and it grabs the moth with its pouch, brings the moth up to its mouth, and it just starts munching on it midair, and so that it can start eating it. Pretty amazing. So the auditory cortex in these animals is really complex, and it's highly specialized to process echoes. So we're going to talk a little bit about how the mustache bat cortex is organized. So we're going to have to think back to one of the earlier lectures when we learned about the auditory system. So what the bat does is that it makes a series of very short calls with the most dominant frequency at 61 kilohertz. So bats typically will be making their vocalizations well above human hearing. So when you see bats flying overhead, sort of at dusk in the summer, you'll see, if you just look up, you'll see all kinds of bats flying overhead. You don't really hear them, and that's because the sounds that they're making are well above what you can hear. And you can also see that these vocalizations are super short. Look at the scale here, milliseconds, okay? So these vocalizations are less than, well, a little bit more than 0.01 milliseconds. And so they're making these pulses of vocalizations super fast. They're going, but like 1,000 times faster than that. And most of the vocalization is at one particular hertz. It's at precisely 61 kilohertz. So one thing that is super interesting about this is that the primary auditory cortex, it's organized tonotopically, just like the human cortex, as we learned about, that there's going to be one part of the human cortex that's going to detect low frequencies, and another part that's going to detect high frequencies. All mammals that have ears and use them, they have this organization. But they have a weird part of their cortex. There's a huge chunk of their cortex, like 1/3 of their cortex, that's dedicated to just 60 to 62 kilohertz. So it's highly specialized for the dominant frequency that they emit. Super interesting. This is because this part of cortex is doing some precise calculations, and you need to dedicate more space of the brain to doing something and focusing on the fine details and processing the information that's coming in order to get the most information out of it. So these bats, they don't really rely too much on the low frequencies or the highest frequencies that they can detect, which is 100 kilohertz, way above human hearing. Most of what they need to be focusing on, since this is how they actually kind of see the world, is using their vocalizations and listening to the echoes. And so it's going to be around 60 to 62 kilohertz. And so this essentially acts like a fovea for hearing in these bats. Just as our eyes have a certain space where we're going to have focus in the highest detail of vision, we don't need high specialized visual resolution for our peripheral vision. We just need to be able to see sort of in that direction. These bats, they have dedicated a huge chunk of their cortex to a particular range of the frequency that they use for doing echolocation. So the mustache bat uses information from moving objects in order to calculate that it is moving, where it's moving, and how fast it's moving. And what it uses to do this is what's called the Doppler shift. What is the Doppler shift? So the Doppler shift, all of you are familiar with this phenomenon. When an object is making noise, say a car is running, let's say you're in a car, like a race car, for instance, it's going to be making a noise. And that noise, as far from your perspective, is going to be pretty consistent. It's going to be right, like you're driving, you're in your race car and it's buzzing along, and it's going to be making that noise. But if you were and so that, or if a race car is just sitting there, that's all the noise that it's going to be making, right? Like, so if you're standing here and there's a race car right next to you, you're going to be hearing a frequency that's not really going to be changing. However, if you, let's say we go over here to this race car that's moving, the pitch that we hear, and we're the red dot, my little pointer here, as the race car goes by, the wavelengths in the front of the race car are all compressed because it's moving. And then as it moves away, the wavelengths are spread apart. So the distance between those wavelengths is greater than what it would be if it was stationary, which means its pitch is lower. So it goes from high pitch to low pitch as it's moving. That's called the Doppler shift. And you've all heard this. So like this race car going by is going to go, it's going to go right. That's what it sounds like. You've all heard this. That's the Doppler shift. That thing that happens as the pitch goes from higher pitch and then goes to the actual pitch and then eventually goes down to lower pitch. So what the bat is doing is that they're able to take this information of a moving a moving moth that's moving in space, and they can detect the shift in the echo frequency that's coming back. And so if a moth is coming towards the bat, for instance, then the echo will be shifted to a higher pitch. And if the moth is moving away from the bat, then the echo will be lower pitch than what the bat actually emitted. And it can use this information to do this. So this echo from the bat's going to be shifted because the bat is flying and the insect is flying. And the bat knows how it's flying, and then it's going to see the relative change with the insect as well. So the bat uses this information to calculate the direction and the speed of the flight path of the insect. There's the main fundamental frequency, which is FM2. If you remember from the previous slide, there were a few additional, so that's a bit of a harmonic stack with a couple of fundamental frequencies, and it calculates. It has areas of its cortex that calculates the difference between all of them, but the Doppler shift area is primarily going to be driven by this part here, and it looks for like a shift in the actual frequency. So the echo, in this case, the echo is slightly higher than the sound that was emitted, which would indicate that the moth is coming towards the bat, relatively speaking. And then there's, of course, a little bit of a delay, right? Because the sound has to be emitted, and then it has to be detected as it bounces back. So it's using this phenomenon of not just sending out echoes and then listening for them to come back and waiting for the time difference. It's also listening for a change in pitch and allowing it to detect the precise location of where the moth is. And we've learned an awful lot about how the auditory cortex processes this kind of auditory information by studying the auditory cortex of bats. So the next example from neuroethology we're going to talk about is the star-nosed mole. This is an animal many of you probably haven't really heard of. It has a specialized sensory organ at the tip of its nose. That's how it gets its name. So this is the star of the star-nosed mole. And this is the range of the star-nosed mole. So it is found in our area, generally speaking. There's not like a big number of them, but it's mostly restricted to these areas up here. It lives mainly in marshy wetland areas, and it's typically found underground in tunnels. It's a mole, right? So it uses these big old claws to dig holes, and it's looking for things to eat underground. So the star-nosed mole, we're going to talk about the star of the star-nosed mole. Why the heck does it have this crazy facial feature right on the front of its face? So it has this star. Obviously, there must be some reason to have a star. And the reason is that it's a specialized organ that is used for touch. It's quite specific. It has 11 appendages, and so we can number them starting from the top, going all the way to the bottom, 1 through 11 on one side, and then we have 1 through 11 on the other side. So a total of 22 on both sides of the nose. And then here are the nostrils here. And if we zoom in on these organs, what we can see is that they are loaded with these specialized touch receptors, these mechanoreceptors. And it's covered with hundreds of these very fine touch sensory organs. These specialized types of touch organs are called Eimer's organs. And if we look inside one of these Eimer organs, what we see is that there are free nerve endings across many layers of dermis. There's a Merkel cell that's in here, and there's a laminated corpuscle in here. So we got all kinds of different types of touch that we can pick up from this appendage. And you can see that it's actually raised. So you can imagine that like getting knocked around like this would also induce certain changes as well. So since this is neuroethology, it's important to sort of take the comparative approach. So we're going to be comparing a little bit of the star-nosed mole, which is one species of a group of moles, but it has this weird, unique feature, right? It has this weird star appendage on the front of it that has this apparently very specialized touch receptors. We can compare it to a closely related species called the eastern mole. So the eastern mole, you can see that there's some similarities. They both have these huge claws because they both are digging holes and looking for prey items. But this is the nose of the eastern mole, and you can see it looks nothing like the star-nosed mole. What's up with that? Why are they so different? Well, we can start by just kind of looking at eating. One thing that these animals spend a lot of time doing is eating. And the fact that they're digging around in tunnels, perhaps there's something unique about the star that's related to food. And that actually is the key to this. So the star-nosed moles, they hunt for very small food items that most other moles really won't pay attention to. If we look at the dry weight of sample prey of the eastern mole versus the star-nosed mole, so they're in slightly different habitats, right? The star-nosed mole is in super wet habitats. The eastern mole habitat isn't quite as wet. I mean, you can even see the difference here, right? Like this is just sort of dirt, and you can see that this is like swampy bog stuff. When you look at the kinds of prey items that are around in the star-nosed mole habitat, and you weigh them, they're really, really small. Less than 1 milligram, upwards to 5 milligrams. The vast majority of their prey items are that small. Whereas the eastern mole has a lot bigger things that it can hunt for. And if you just do the average prey mass of the star-nosed mole typical prey item versus the eastern mole, it's a huge difference. So the average star-nosed mole prey item is probably around the order of less than 10 milligrams, whereas the typical prey item for the eastern mole is around 140 milligrams. So it's almost a tenfold difference. It's more than a tenfold difference between these two. So perhaps this has something to do with it. The other thing that we can look at then, is the efficiency of hunting for items, right? Like if you are out looking for food, and let's say you need 1,000 calories to eat. You could go down to Lefties, that's on South Main, and you can order a cheeseburger and fries, have a nice soft drink alongside it, and at one sitting, boom, you've got more than 1,000 calories. You're good to go with that one sitting. That's essentially the strategy that the eastern mole habitat is. The eastern moles are doing. They're finding relatively big things like crickets and earthworms, and they're getting a lot of energy out of it. So instead of calories, we're just converting it to joules, just another form of energy. And but you know what? Like, all right, you got to go down to South Main, and you got to go into Lefties, and you got to wait in line, and okay, so it's going to take a little bit of time, and then you order your burger, and you got to wait for it. It's going to take some time to do this. The same is true for the eastern moles when they're hunting for crickets and earthworms. Like these things aren't too common. They got to find them and dig around them. They got to catch the cricket. It takes some time to deal with these things, and this is called handling time. And so it's not super efficient as far as time goes, but because they're so big and you get a lot of energy out of them, the reward is quite good. So if it only takes them around 20 seconds to eat a cricket or an earthworm, the profitability, how efficient we are being with getting our prey items, is quite good. So if you go into Lefties and you get your burger within 10 minutes, boom, that's pretty good. You got your 1,000 calories very quickly. But sometimes it might take 50 minutes or an hour because it's a Friday and you got to wait to get a table. So now our efficiency is a little bit lower. And so our profitability is a little bit lower, but we're still doing okay. You still get your 1,000 calories at the end. And, there's just going to be some sort of relationship to how much time you have to waste finding these things in order to get them. When you talk about small prey items, you're not getting a lot out of them. You're only getting 10 joules of energy out of each item. Really, really small items are not going to be very energy efficient. This is the equivalent of like going across the drill field and someone has scattered Ritz crackers all over the place, and you got to go hunting around and you find a Ritz cracker and eat it. And it might take you an hour to find all those Ritz crackers because it's kind of stuck in the grass and you can't, and maybe covered up with leaves. Not a great way to eat. You'd rather just go to Lefties and wait 45 minutes to get a burger. So how can the star-nosed mole be efficient with these small prey items? The key is that they have to find them quickly. If it takes them 20 seconds to find a tiny little item, the profitability is super duper low. they need to be, in order to do this, they have to be able to find these small prey items efficiently and quickly. And so the handling time can be efficient, up there with the earthworm and the cricket, if they're able to eat them within a second or less.
So how is it that the star-nosed mole is able to take advantage of these tiny little food items? Well, let's start by just looking at the behavior itself. So this is a video, real-time video, of a star-nosed mole moving around and finding a tiny little food item that's been placed on a piece of glass. So the mole is on top of the glass, and so the glass is like underneath. It's standing on a glass sheet, and these researchers have outfitted a tiny little bit of this food onto the glass. And what you can see is that the mole is like moving around like crazy. So you can see its handling time is pretty fast just because it's moving very fast. And it's just like, there it is, and now it's gone. So what's actually happening when the star-nosed mole is doing this? Well, it turns out that the star-nosed mole is making a saccade to the food item. And so we will take a look at what that looks like in slow motion. So let's take a look real fast in real time what this looks like. This is a real time, right? We just saw this, grabs the food item. This is what it looks like in slow motion. So watch what's happening with the star-nosed mole. So this is 1/50th slowed down. And you can see it's with these appendages, it's just moving them around, searching the environment. This is the same video we just saw, and it can smell that there's something over in this area, but now it's trying to find the food item. So it's feeling around with the star. So the star allows it to see as much space as possible. And there, it's touched it. Now it orients to this, and then watch what happens. It reaches out with like these weird little teeth and grabs the food item and moves on. Pretty amazing that it does that. So the handling time is very fast, and it's searching super efficiently with the star because it has this wide touch organ that it's just touching around looking for food. And then it can make a saccade, basically like orienting the star directly to a central place and then grabbing the food item is exactly what you just saw. So it's specialized in so many different ways. Those crazy teeth that you just saw that sticks out between the star, that's where its mouth is. So the star actually covers up its mouth, and it has these highly specialized incisors that are really precise. They're almost like tweezers for grabbing tiny little food items. So it grabs a little food item and then just eats it and swallows it super quickly. And it reaches out between the star right at the middle. So let's compare what this is like with the star-nosed mole versus the eastern mole. So let's look at the star-nosed mole again in real time. That's 10 food items that it grabbed in a second, maybe not even. And now we're going to look at the same thing, but the same kinds of food items for the eastern mole. Remember, the eastern mole really doesn't eat these kinds of things. It tends to eat crickets and worms and that kind of thing. So this is like just too small for it to really care for. So let's take a look. Ten food items. It smells it. It's like, yeah, there's some food here. I got one of them. You can see it spent like 5 or 6 seconds, and I think it ate this one. And I think this one just ended up on the bottom of its belly because it was just too small to really handle. So you can see that the star-nosed mole is super efficient at hunting down these kinds of food items. It ate those 10 food items like it was nothing, just boom, 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 boom. And with these 10 food items, the eastern mole got one of them, I think, and the other one just ended up on its belly, and it didn't even really bother and just continued on because it's just too small for it to really bother with. So there's some really interesting things that are true about the star-nosed mole brain. Obviously with having this specialized sensory organ, it won't be surprising to hear that its representation in the somatosensory cortex is quite elaborate. And in fact, each of these appendages, the 11 appendages, you can outline them on the cortex and within the other parts of the sensory nervous system, like the trigeminal nucleus. So, which is like where the facial nerve comes in and where you're going to get somatosensory information from the trigeminal nerve. You can outline the each appendage of the star just within the trigeminal nerve itself. Goes through the thalamus, the VPM, as we learned about, right? Goes from the trigeminal nerve up to the thalamus. And then this information is taken up to the cortex, the somatosensory cortex. And then if you look at the details of the cortex, this is what the somatosensory map of the sternal cortex looks like. The star itself is found on, this is on either side of the brain. It takes up around something like 50% of the somatosensory cortex, and each of the appendages of the star gets representation. Interestingly, when you look at the overall map of the cortex, that this appendage, which is relatively small compared to some of the other appendages, is actually quite large in the sternal map, and we'll see a little bit more about that in the next slide. But this is essentially where the fovea for the star is, and you can see when the star nose mole makes a saccade to this tiny little food item, it basically tries to figure out the precise location of the food item using this appendage. 10 and 11 really are the main parts of it. So you can see that nearly 2/3 of the somatosensory cortex is dedicated to the 9 appendages, whereas 1/3 is dedicated to just 2 of them. And then this is what the star-nosed mole homunculus looks like. So they use their forelimbs for a fair amount, right? They've got these giant forelimbs, the claws for digging in the ground. The trunk and the tail and the hind limb are relatively small, but the star takes up a huge part of it. And so if you were to draw out what the star-nosed mole homunculus looks like, it's going to look like this. Pretty cute, if not terrifying. So the area of cortex, just as we were looking at that tiny little panel, that's dedicated to each of the appendages, the rays here is what they're calling it in this figure, it's emphasized on the fovea. You can see that 10 and 11 take up a huge part of the somatosensory cortex, and 11 in particular. And so this is going to be that highly specialized organ. And then, so just like our vision, the star has a periphery, and that's what rays 1 through 9 are. It's the peripheral vision. They're trying to find tiny little food items and getting a sense of it. And then they get precise information of where it's located, and that's in the fovea of the star, which is going to be appendages 10 and 11. And you can see that the somatosensory cortex is more highly dedicated to this part, just like it is for the quote-unquote fovea of the auditory cortex in the echolocating vertebrates, as is the fovea in humans, in the visual cortex, the same thing. We can also see that the reason why it gets more space in the somatosensory cortex is because it has a higher density of Eimer's organs. Just like our fovea in the visual in our eye, we have a greater density of cones, and we have very little convergence, right? Almost every single cone in the fovea and the rods that are in the fovea are basically one-to-one representation with cones to to retinal ganglion cells. And the same is true for the specialized rays 10 and 11. They have a greater density of Eimer's organs to give that specialized location of where that small little item is, and then they can quickly find it. So that's it for the star-nosed mole. Now we're going to talk about the last example, which is the tentacled snake. So this is a snake that's found in Southeast Asia. It's a fish-eating aquatic snake. And generally what it does is that it rests on, it tends to live in sort of like swampy areas where there's like a lot of branches and trees that are basically overlaying and kind of sticking into the water. And the tentacled snake sits there in the water and hunts fish. And so we're going to learn a little bit about how it does that. It's called the tentacled snake because it has these weird tentacles at the front of its face. The tentacles are sensory organs as well, and it detects vibrations of fish. So as fish are moving, they're going to create small waves that will propagate from the fish and make its way to the snake. The snake, of course, also uses its eyes to do this, and so it has sensory information, somatosensory information, visual information coming in. This information is processed by what in snakes is called the optic tectum. The optic tectum is the superior colliculus in snakes. It's the same thing. And so this is a basic somatosensory and visual map in the superior colliculus, as we learned about in the visuomotor portion of the course. So it's the same kind of map. And what the snake is using then is going to be taking this information to get precise location of where the fish is so that it can strike. This is what it looks like. So the snake senses the fish is there, and it's just hanging out, sort of pretending like it's a branch. And you can see that this is all in slow-mo, obviously, slow motion. And the snake just kind of moves and grabs the fish. Now, you should go back and look at this slide again and watch that video again, because there's something really interesting going on there. And I want you to watch it again and try to see if you can pick up on what's going on. So go ahead, watch it, and then come back to the next slide. So one thing that the fish use is a specialized escape system. It's called the Mausner escape system. And this allows the fish to make what's called a C-start in order to escape predators. So the fish, it also has the ability to detect vibrations. So small waves propagating towards the fish are sensed by the somatosensory organs on the side of the fish. And then it has this specialized system. So it kind of acts like ears. It's not exactly ears, but we're going to call them ears here. What happens in this case, if the sound is coming from this side, it's going to be detected by these specialized cells that feed into these huge neurons called Mausner neurons. And so you're going to get activity that will cause these neurons to fire, and then it's going to go down into this circuit. So the activity goes down into the Mausner circuit, where what it does is that it it branches off into a couple of places. So it crosses the midline, just like it does like in other vertebrates. We have the afferents crossing the midline, and then it branches onto two different neurons. It branches onto one neuron, which is an inhibitory neuron. So it's going to cause that one to fire. It also branches onto an excitatory neuron, some muscle cells, that will cause the muscles on the right to contract. And then it's synapsing onto these inhibitory neurons that inhibit the opposite side. So that crosses the midline and inhibits these motor neurons so that the firing goes down. So you have these muscles relaxing, these contracting, and it's going to cause the fish to bend and turn away from the sound. So it hears the vibration, senses the vibration. The Mausner cell on the left fires because it's the one sensing it, and it causes the muscles on the right to contract, the muscles on the left to relax, and since they work in opposites, then it allows the fish to turn like this and escape. And this is an escape system. It's intended to be fast. The fish can do this within 25 milliseconds of sensing the sound. So it's a super fast instinctual thing that fish will do. They don't even think about it. I mean, fish don't do a lot of thinking anyway, but they rapidly will respond and move away. So let's take a look at what's happening with the C-start. And I want you to pay attention to the snake and try to observe what happens to the fish before the snake actually strikes. And watch what the snake does to get the fish to move. So you see the fish basically does a C-start and it does a C-start directly into the snake's mouth. So what's going on there? Same thing. If you noticed, the snake starts to move its body here, causing the fish to react. And then the fish basically does a C-start and swims directly into the snake's mouth. Move, eat the fish. So the snake is taking advantage of the fact that fish do these C-starts. It's tricking the fish into doing a C-start and then swimming directly into the snake's mouth because it knows it's going to make this instinctual C-start. It's going to curve its body and then tricks the fish into swimming directly into its mouth. And it does this by moving its body before it moves its head. So this is basically a diagram of what this looks like. The snake makes a small feint, so it's feinting with its body to trick the fish into swimming into its mouth. So it creates these waves that come from the body of the snake. The fish senses this, and then the snake will move his head right after moving its body. The snake is basically making a calculation depending upon the position of the fish in order to turn its head so that it'll eat the snake. So you can observe this here. So here's the fish. Here's the snake has moved his body. You can see the body has moved and he's starting to move his head. And then the fish he's doing a C-start, swimming directly into the snake's mouth. Here's another example where the fish is in a slightly different orientation. So the snake has to sense this. Moves his body. You can see the body is curved out here, and the fish is starting to do a C-start. And the fish swims directly where the snake is going to strike. And so it doesn't work perfectly every time. I mean, they do miss. And so here's a fish over here. The snake has made a movement, and the snake has predicted that the fish would swim this way. But it turns out, in this case, the fish, for whatever reason, turned the other way, and so the snake missed. So the gray here is what the snake was predicting. You can see it didn't even try to get the fish. It just moved to where it thought it was going to move, and the fish just happened to go the opposite direction. So it missed. And that does happen from time to time. So that is it for this lecture. I wanted to make this relatively short. I wanted to also say how much, challenges of doing a class like this online, and that it would be so much better to do in person. But it is what it is, and hopefully things will get better for 2021, and then I hope that you've gotten a lot out of this class, so that you have a better appreciation for the brain and how it controls behavior and the various systems of the brain, because it is an amazing part of biology, and we're very lucky to be able to study it. So in this course, for this lecture, we're talking about neuroethology. You should know the definition of it. What are some of the major examples of neuroethology that we learned about in this class? What is echolocation and how do bats use it to hunt, and what's specialized about their auditory cortex? For what is the star and the star-nosed mole used? How do they use it? Why is it specialized in this species than in other species of moles? What are some of the key features of how the star is represented in the nervous system? How does the Meusner escape circuit work in fish? So this is an interesting circuit that you should have some knowledge about. And then how do tentacled snakes take advantage of this escape response? And so that's it. Take care, stay safe, and we'll talk to you later.